Hello everyone, my name is Lanney, and me and my partner Amon will be presenting FuncFetch, an LLM-assisted workflow enables mining thousands of enzyme-substrate interactions from published manuscripts by Smith et al. So just to illustrate this problem, I'm here on the Uniprot website, which is one of the largest freely accessible databases for protein sequences and functionality, among other things. The authors of this paper created a manually curated dataset of the enzyme BAHD acyltransferase to evaluate FuncFetch against. So we'll use that as an example. So if we perform a search for all genes that encode this enzyme, we get over 1,600 results. And if we examine the first result here, we can see that this particular gene that encodes the enzyme has been manually reviewed and annotated, and has an annotation score of 5 out of 5. So we can see that the amino acid chain is 435 long here, and we can clearly see that its functionality is present with citations. We can see that we know its active sites, so it has 2 proton acceptors and the location within the amino acid chain. We can see further documentation of its functionality here with citations again. We can also see its location within the cell. So in this case, the enzyme is found in the cytoplasm. And we can also see its AlphaFold 3D predicted structure here. So as we can see, in this case, we were able to get a very clear overview and understanding of this particular enzyme, of its functionality, its active sites in this particular organism, which is watercress. Yeah, mouse ear cress. And this is thanks to the work of biocurators, curators, who must manually find the related works for these particular enzymes' functionality, review it, cite everything properly, and then input it into central databases like Uniprot. So this is a very time-consuming task because it all has to be done manually. So unfortunately, it is not the norm for most enzymes to be annotated like that. If we go back, remember we had 1,600 results, and of the results here, only 5 have been manually annotated. The rest have been automatically generated, and as we're about to see, there's much less detail available. So if we click on a random one, we can see that the annotation score has dropped to 1, and while we know the amino acid chain length of 449 in this particular organism, we have no detailed information about its functionality. We have no information about its active sites or what they are or where they are. We do not have information about its location within the cell. And while we do have AlphaFold's predicted protein structure here, we basically have no other details about this enzyme's activity. And so, unfortunately, the vast majority of entries are not manually annotated, and so information about them is not readily available like here. That is not to say that the information is not available at all. It might be, but biologists seeking information about an enzyme's activity in a particular organism and where there's no manual annotation available, they will have to go through a time-consuming research process to find its functionality within publications, if it exists at all, or if it doesn't exist, then they'd have to go find out themselves. So basically, our problem is that the number of sequenced genomes is rapidly increasing, but the function and annotations of proteins and enzymes is not keeping pace. Going back to an example on Uniprot, of the 16 million entries in Uniprot under the Viridae Plantae taxonomy class, only about 43,000 have been curated, which is 0.27%, and only 12,000 of those 43,000 have functions or catalytic activities listed. It's a similar case for the Kyoto Encyclopedia of Genes and Genomes, or KEG. There's 22,000 enzyme entries, however, only 12,000 of them have been experimentally verified. So overall, the bottleneck is this biocuration process, which was previously mentioned in the example. It's just extremely labor-intensive and too slow. So when looking to curate enzyme activity and functionality, biocurators must search for related papers in databases such as PubMed or Web of Science, and then they have to manually extract the enzyme activity data. Then, after cross-referencing with other databases such as Uniprot or KEG or Brenda, then they have to structure the data into standard formats so it can be used across multiple databases. Finally, after this, they have to do quality control and validation on their data. And then they can, again, manually upload it to the multiple databases such as Uniprot. So obviously this process is very time consuming, especially considering the scope and sheer number of enzymes it has to be done for. A 2022 report estimated that there was less than 100 full-time biocurators worldwide, which is obviously not enough to keep up with the pace of global genome research. So this further highlights the need for some form of automated extraction of enzyme activity from literature. So just to briefly go over some related works and previous attempts to tackle the problem of data availability and the creation gap, include the paper Named Entity Recognition for Gene Disease Links by Milosevic et al., where the authors found that a PubMed-BERT-based model was superior to rule-based systems at identifying gene disease links. However, their approach required labeled datasets and didn't cover enzyme interactions, which is our area of focus here. In 2024, Rahana et al. Used models like GPT-4 and BERT to achieve high precision in extracting protein interactions from a given paper. However, this only applied to well-studied proteins for which a large body of work existed already. Finally, in 2023, Boadu et al. Used the protein language model and 3D neural networks to predict protein function. And so by combining feature embeddings with protein structures predicted by AlphaFold 2, they were able to outperform several state-of-the-art methods. So to tackle the problem of the curation gap between the increasing number of available sequence genomes and function annotation, the authors created FuncFetch, an LLM-powered solution to automate enzyme extraction from literature. Just to give a brief overview of how it works before Amon explains in detail, first PubMed is queried with a set of user-defined keywords, and then the returned abstracts from PubMed are filtered for the presence of the desired enzymes. The PDFs of these filtered results are then downloaded and have their enzyme activity extracted using GPT-4, and then these are further automatically curated and filtered using a Python script to produce a dataset which can then be further manually curated by the end user. This method results in much faster curation, better database completeness, and lower manual effort, since the end user can curate an already filtered dataset generated by FuncFetch instead of going through individual papers to look for enzyme activity. And now I'll let Amon explain the specifics of how FuncFetch works.